Hello, I'm Dr. Charles Flexner from Johns Hopkins University in Baltimore, Maryland. And for the next 30 minutes, we're going to be talking about new antiretroviral therapy, including drugs and formulations. These are my disclosures as required by the organizers of the meeting. At present, in late 2020, there are more than 20 different agents in development for treatment and prevention of HIV. That's especially true if you consider the large number of broadly neutralizing monoclonal antibodies, or bNAbs, that are being developed by a variety of agencies. Obviously, in 30 minutes, we could not possibly cover all of these agents. And so I'm going to focus on the small molecules and long-acting formulations that are in the most advanced clinical development. Let's start with small molecules being developed largely for heavily treatment-experienced patients with multi-drug resistant HIV. These agents have a number of targets, mostly in the interior of the cell, designed to prevent the maturation of products of the P24 antigen gene, including the capsid protein. And there are several of these small molecules in clinical development currently. That includes Beviramat, which is in phase 2, but has shown that about half of treatment-experienced patients had no response due to polymorphisms in the GP120 envelope gene. GSK 3532795 in phase 2B, which has produced a significant drop in viral load in the majority of patients, 76 to 83%, but was associated with gastrointestinal intolerance. GSK 2838232 in phase 2A has produced a 1.7 log average drop in viral load at day 10, but requires boosting. And finally, GSK 3640254 is about to begin phase 2 testing. All of these agents are oral, and at present, all of them need to be given at least once per day, if not more frequently. On the other hand, there's substantially more activity and perhaps more excitement about long-acting formulations being developed for treatment and prevention. And I'm going to start with long-acting cabotegravir and long-acting rilpivirine. These two studies have presented the phase 3 results demonstrating safety and efficacy of this two-drug combination in individuals previously taking oral antiretroviral therapy. The so-called ATLAS study was carried out in antiretroviral experienced patients who were randomized to continue their daily oral therapy or switch to a 4-week run-in of oral cabotegravir and rilpivirine to assure tolerance, followed by a switch to 48 weeks of injectable cabotegravir and rilpivirine as their sole antiretroviral treatment. The FLAIR study had a nearly identical design, but took place in treatment-naive patients who received 20 weeks of a co-formulated daily oral dolutegravir-based regimen before being randomized to the all-injectable regimen or continued oral drugs. The ATLAS study showed that the injectable regimen was non-inferior to the continued daily oral regimen, whether one looked at the number of patients failing therapy or the number of patients in whom continued therapy was successful. And that's shown in this slide. Now, unfortunately, there were some cases of resistance to rilpivirine and cabotegravir in these registrational studies, although resistance was rare. There were 3 cases reported in the Atlas study. All 3 of these patients had resistance to rilpivirine, with 1 having resistance to both rilpivirine and cabotegravir, and all 3 cases occurred in individuals with subtype A or a hybrid AG virus. These are viruses that are most common in Eastern Europe and in Central and Eastern Africa. In addition, the plasma cabotegravir and rilpivirine concentrations at the time of failure were below the population means for all 3 of these patients, but within the range for the large majority of individuals who maintained virologic suppression. And thus, most of the people who had drug concentrations on the low end actually had successful therapy with cabotegravir and rilpivirine. Now, the FLAIR study, this was the study in treatment-naive individuals, had nearly identical results. That is, the primary endpoint showed that the long-acting therapy was non-inferior in terms of the number of patients with viral load undetectable at week 48, viral load detectable at week 48, and similarly with the key secondary endpoint, the number of patients who had undetectable viral load at week 48. But like the Atlas study, there were 3 cases of resistance in the FLAIR study, and again, 2 of these 3 patients had resistance to rilpivirine, and 3 had resistance to cabotegravir. All 3 had subtype A virus at baseline, and also had low concentrations as compared to the low drug concentrations as compared to the population means. Now, the Atlas study had an extension which changed the administration frequency of the long-acting injectables from once every 4 weeks to once every 8 weeks. And so at the end of Atlas, participants were offered the option of switching to this extension arm, in which they were randomized to either every 4-week cabotegravir and rilpivirine or every 8-week cabotegravir and rilpivirine. And at the end of 48 weeks, the every 8-week regimen was non-inferior to the every 4-week regimen based on either the primary endpoint, that is the number of virologic failures, or the key secondary endpoint, that is the number of patients with an undetectable viral load. It's important to point out, however, that when the treatment failures on Atlas were evaluated, there were 8 patients with confirmed virologic failure on the every 8-week regimen and 2 on the every 4-week regimen. 6 of these 8 on the every 8-week regimen had rilpivirine resistance mutations. And so although resistance was still rare, there was more resistance on the every 8-week regimen than on the every 4-week regimen. So cabotegravir and rilpivirine given every 4 weeks or every 8 weeks were non-inferior to standard of care antiretroviral therapy in 2 phase 3 clinical trials. Resistance to these drugs was rare, but mainly seen in patients with subtype A virus. Also, most of these patients had the L74I polymorphism in the integrase gene at baseline. All patients had cabotegravir concentrations in the lower quartile, and resistance was more common with the every 8-week regimen than with the every 4-week regimen. Only a single patient developed resistance because of discontinuation of therapy, and all of these treatment failures with resistance occurred in people who showed up regularly for their every 4-week or every 8-week injections. This 2-drug combination has now been approved for prescription use in Canada, has been given a favorable review by the European Medicines Agency, and is under review by the FDA in the United States, with approval expected sometime in 2021. Now, that was treatment with cabotegravir and rilpivirine. What about prevention with these long-acting formulations? Well, I'm going to talk to you about the results of a large randomized trial conducted by the HIV Prevention Trials Network, in which a long-acting injectable formulation went head-to-head with a 1-pill once-per-day PrEP regimen for HIV prevention. And this was the HPTN 083 study presented this past summer by Raphael Landowitz from the University of California at Los Angeles. This study took about 4,500 men who have sex with men and transgender women and randomized them to an arm of injectable cabotegravir for PrEP or a daily oral tenofovir and emtricitabine for PrEP. However, this was a double-blind, double-dummy trial. So every participant was asked to take pills every day, and every participant was asked to come in and get an injection every 8 weeks. The injection would either be cabotegravir or cabotegravir placebo, and the pills would either be tenofovir emtricitabine or tenofovir emtricitabine placebo. The length of the study was based on the number of events, that is HIV seroconversions, and the study was stopped by the Data Safety and Monitoring Board after an average on-study duration of nearly 2 years. So there were 52 HIV infections, 1.4 years median participant follow-up, and the final analysis showed that the risk of failing PrEP was about 3 times as high in the daily oral arm as in the injectable arm. So there were only 13 infections in the injectable cabotegravir arm and 39 infections in the tenofovir emtricitabine arm, a highly statistically significant outcome. Following the first 6 months of the study, there was a continual divergence between the oral arm and the injectable arm, suggesting that the benefit of injectable therapy was maintained throughout the duration of the study. Now, in addition to this large study in men who have sex with men and transgender women, there was also a large randomized prospective study carried out by the HIV Prevention Trials Network in cisgender women, most of whom had a heterosexual risk factor for HIV infection. This study enrolled about 3,200 women, and the study design was identical to that of the HPTN 083 study, that is a double-blind, double-dummy, so everybody took pills and got an injection every 2 months. But one arm received only injectable cabotegravir as their active PrEP, and the other arm received only oral tenofovir and emtricitabine as their active PrEP. The DSMB asked for unblinding in November of this year because of an unexpected difference in efficacy in the 2 arms. And that difference favored the injectable arm. In fact, it greatly favored the injectable arm. When the study was stopped, there were 34 HIV infections in the daily oral tenofovir emtricitabine group, and only 4 infections in the long-acting cabotegravir group. Again, a highly statistically significant difference, and about 9 times the infection risk with oral therapy as compared to daily every 8 weeks injectable cabotegravir. I think there are some important implications of the HPTN 083 and 084 studies. The first is that one drug, cabotegravir, was actually better than 2 drugs, tenofovir and emtricitabine, for HIV prevention. At this point, it's unclear exactly why that difference occurred. One issue is the better pharmacokinetic profile over time of the injectable cabotegravir regimen, such that drug concentrations were maintained in the vast majority of study participants above the targets necessary to prevent infection with HIV. It also appears that there was non-adherence to daily oral PrEP, not unexpectedly, but it's possible that the difference between non-adherence with daily oral PrEP and the coverage of injectable cabotegravir every 2 months explain this resistance. It's also true that cabotegravir has a higher barrier to resistance than tenofovir or emtricitabine, and that may have played a role. But it could be something else that still remains to be determined. And I think we're going to learn a lot more about this in the coming 12 to 18 months. Regardless of the cause or reason, this is a clear demonstration of the benefits of a long-acting formulation as compared to standard one pill once per day antiretrovirals for prevention. I'm going to move on now and talk about the next new long-acting small molecule, and that is Lenicapavir, a capsid inhibitor developed by Gilead that is administered subcutaneously, unlike cabotegravir and rilpivirine, which are administered intramuscularly. So how do capsid inhibitors work? Well, this capsid inhibitor is a small molecule that blocks maturation of the HIV capsid protein, but also interferes with its translocation in the nucleus, thus interfering with HIV integration once the virus gets into the cell. So the drug has several distinct mechanisms of action which may contribute to its potency and efficacy. The drug does have an exceedingly low EC50 in peripheral blood mononuclear cells. It's active against all HIV subtypes. The resistance variants that do emerge in vitro have very low fitness and therefore may have a difficult time propagating in the clinical setting. The drug also has very low clearance and solubility such that it has a very long half-life when given subcutaneously, a half-life in humans of 30 to 43 days. Both oral and subcutaneous formulations are in development. The oral formulation mainly to assure tolerability, but also to allow the company to conduct drug interaction studies. The drug has very potent anti-HIV activity in short-term 10-day monotherapy studies, shown in this figure, at doses ranging from a single dose of 20 milligrams to a single dose of 450 milligrams, with up to a mean of 2.2 logs, a decrease in a viral load at 10 days. As potent as the activity in humans of any other antiretroviral monotherapy. And phase 2 studies with this drug are in progress. But the drug is a long-acting injectable formulation. And this study looks at the pharmacokinetic profile in HIV or HIV seronegative individuals given 3 different single subcutaneous injections of lenacapavir, ranging from 300 milligrams in the blue to 900 milligrams in the gray and the yellow, either given as 3 1 milliliter subcutaneous injections or 2 1.5 milliliter subcutaneous injections. And what you can see from this pharmacokinetic profile is that the drug reaches and maintains target anti-HIV concentrations, in this case, 6 times or greater the inhibitory quotient for in vitro inhibition of HIV. And those levels can be maintained for at least 6 months, 24 weeks in these small phase 1 studies. But that suggests that this drug could be given on an every 6-month basis as a subcutaneous injection. We'll move on now from injectables to implantables. And we're going to talk about Islatrovir, a new anti-HIV drug that is also being developed as an implant. So why would one want to develop an implant in the first place? Well, implants have potential advantages over injectables. That includes the fact that they are removable. That is especially true for the inert or early bioerodible forms after they are placed in case of adverse events. They provide more consistent and predictable drug release than injectables, particularly intramuscular injections, and the pharmacokinetics are therefore not dependent on the injection site. Some of these implants can remain in place for years. The inert, non-degradable subcutaneous contraceptive implants, for example, may remain in place for 3 to 5 years before they need to be replaced. But there are some potential disadvantages over injectables. There is a specialized device required for insertion. There is a minor surgical procedure required to remove these devices if they need to be removed. At the end of their usable lifespan, that is, when they run out of active drug, they need to be removed and replaced because one cannot tell by palpation whether or not these implants still are releasing active drug. Their regulation is more complicated than that of injectable long-acting formulations because they're regulated as both a drug and a device. And that may introduce some difficulties in moving these drugs into a generic marketplace, as would be required for use in most low and middle income countries. Now, there is a lot of experience in the field with implantable drugs, particularly in the field of hormonal contraception. Over 40 million women worldwide now use injectable contraception, and nearly half of modern contraception users in sub-Saharan Africa rely on injectable contraceptives to prevent pregnancy, as opposed to oral contraceptives. However, returning to a healthcare provider for an injection every 2 to 3 months has been considered a disadvantage of DMPA, methoxyprogesterone acetate, which is the most common injectable form of hormonal contraception worldwide. And the discontinuation rates of injectable contraception in sub-Saharan Africa are therefore high. And that's contributed to the growing popularity of longer-acting implants. There are several of these now available in a generic format. For example, Norplant 2, which is levonorgestrel, is available as Jadel, Levoplant, or Sinoplant. These are 2 small rods which are implanted once to deliver 5 years of effective contraception. And that 2-rod implant is now being used by millions of women in sub-Saharan Africa. The cost is surprisingly low. The cost per generic implant is less than $15 US, or about $3 US per patient per year for effective hormonal contraception, making this by far one of the most cost-effective means of contraception currently available. So now along comes Islatrovir. Islatrovir is also known as EFDA. It is a deoxyadenosine analog. It has a unique mechanism of action as an nucleoside reverse transcriptase inhibitor. It does terminate the DNA chain like other NRTIs, but it inhibits reverse transcriptase by preventing translocation of the replication complex. And that may lead to some of its unique anti-HIV properties. It also has a very long half-life, 50 to 60-hour half-life for the parent drug in the plasma, and a half-life well over 100 hours for the active metabolite, which is the nucleoside triphosphate inside human peripheral blood mononuclear cells. The drug is exceedingly potent, with an EC50 of about 200 picomolar and broad HIV coverage in terms of strains, including multi-drug resistant strains. There is a low-dose oral formulation as well as parenteral formulations in development, and the drug is currently entering phase 2 studies as an oral drug, either in combination with other agents for treatment of HIV or as a standalone agent for HIV prevention or PrEP. The drug has very potent anti-HIV activity when given in these short-term 10-day monotherapy studies. And this is a figure looking at the drop in HIV viral load in patients not taking other antiretrovirals who are given doses of islatravir as low as 0.5 milligrams as a single oral dose. Now, the high potency of islatravir also makes it a good candidate for formulation into an implant. And because the folks at Merck who are developing islatravir also develop implantable Nexplanon, which is a hormonal contraceptive implant, have used that same technology to develop islatravir implants for HIV prevention and possibly eventually for HIV treatment. And so the islatravir implant uses the same polymer, the same design as the Nexplanon implant, which is an etonogestrel implant. It is a non-bioerodible, removable implant, and it uses the same applicator as Nexplanon, which is shown here. And any healthcare provider can be trained to administer this implant subdermally. This is the simulated pharmacokinetic profile of an islatravir implant as compared to oral islatravir given as a 2 milligram single tablet once every week, showing the implant pharmacokinetics in the blue over 6 months, and the, over 4 months, and the every weekly islatravir in the red. These are actual data of humans who are HIV seronegative, human volunteers, given one of two types of islatravir implants. Either a 54 milligram implant on the left or a 62 milligram implant on the right. And the target for islatravir concentrations is shown here in the dashed line in the blue, and that is a target for several fold above the expected EC90 in peripheral blood mononuclear cells in humans. And so this is not islatravir in the plasma. This is actually the islatravir triphosphate, the active metabolite, being measured intracellularly in human peripheral blood mononuclear cells. And in this particular phase 1 study, islatravir implant is inserted at time 0 and then removed in these HIV seronegative volunteers at week 12. And in both cases, the mean islatravir concentrations are maintained well above target, but you get much higher concentrations with this 62 milligram implant as compared to the 54 milligram implant. A few of these recipients dropped below the target with their intracellular triphosphate concentrations, but none of these recipients dropped below target with their islatravir implant. Once the implant was removed, the decay of islatravir concentrations was as expected based on what was seen with orally administered islatravir. And what this proves is that all of the islatravir in these study participants is coming from the implant. That is, there's no local or distant depot of islatravir formed in these recipients, and thus when you remove the implant, the drug degrades at its expected rate. Now, this is projected islatravir concentrations over time based on those 12-week studies. And based on the 12-week studies, modeling exercises suggest that a 62 milligram implant of islatravir could produce concentrations well above target, that is 0.05 picomoles per million peripheral blood mononuclear cells, which is the HIV prevention threshold. Those concentrations would be sustained for at least 52 weeks a year. And so now we have a practical approach to deliver an antiretroviral in people for more than a year with a single administration. So this is another potential game changer certainly in HIV prevention. And if we could find a partner for islatravir implants, it would also be a game changer for HIV treatment. So in summary, there are more than 20 investigational antiretroviral drugs and biologics in clinical development in 2020. We've talked about just a few of those today, some that I think are some of the most exciting and most likely to make it to drug approval. Most of these agents have exceedingly long half-lives and/or are formulated for long-acting extended release delivery. And in the near future, antiretroviral regimens for HIV treatment and prevention may involve drug administration once every 6 to 12 months or even longer. And so I think this is an exciting part of our future for HIV treatment and prevention, and something we all can look forward to. I want to end now, and I want to thank those who were willing to share with me their slides, some of which included unpublished data. I want to especially recognize Marta Bofito at Chelsea and Westminster Hospital in London, William Spreen at ViiV Healthcare, Roy Gulick at Cornell Medical Center in New York City, Susan Swindells at the University of Nebraska Medical Center, and Raphael Landovitz at the University of California, Los Angeles. And I would also like to acknowledge my funding sources that sustain my interest and research in this area. And also, I would recommend to you, if you are interested in learning more about long-acting HIV formulations, to come to the website for my research resource program, LEAP, the Long-Acting Extended Release Antiretroviral Research Program. Here is our web address, longactinghiv.org. And there will be much more information there for you, as well as links to recently completed studies. So I want to thank you for your attention today. I hope this presentation has been useful, and I hope you are all enjoying this conference. Goodbye.